Okay, so it's my great pleasure to welcome Professor Keith Lindsey. He did his first degree in Oxford at St Catherine's College and then a PhD at Edinburgh University. He did postdocs in Edinburgh and Rothamsted and then became leader of the Plant Molecular Genetics Group at Leicester Biocentre. He had a lectureship at Leicester University before he moved to become Professor of Plant Molecular Biology at Durham University in 1996. And his research works on plant development biology, in particular on molecular control mechanisms and root development and predictive mathematical models. He has collaborative projects throughout the world. He's also the editor of the journal New Phytologist and chair of the board of trustees for the New Phytologist Foundation. So it's my great pleasure to welcome Professor Keith to talk about genes and genetics.
Take the mic, please.
Is it not?
Well, I've got mine on, so can you hear that? That's fine.
Okay. And we'll have time for questions at the end. Thank you.
Thank you very much. It's a real pleasure to be here back in Edinburgh. Thank you very much, Maria, for inviting me to give a talk. She asked that I give a talk on GM, which is not what I normally would be talking about. I've been normally talking about root development. But for 11 years, I was on a government committee called ACRE, Advisory Committee on Releases to the Environment, that advised government ministers on GM and related issues. And so it's an area that I'm kind of interested in, have been for a long time. And what I want to do today is to try to give you some ideas about the current state of GM technology, how it's being used, and some indication of where the technology might take us in the future. So let's see how it goes. Obviously, I don't need to convince anyone here that plants are important. They're the source of a whole range of products from food, fibers, pharmaceuticals, and of course, they're absolutely essential for players in controlling oxygen and CO2 levels in the atmosphere. And of course, agriculture is also extremely important and increasingly so. And that's kind of the theme that I want to develop in this talk this evening. The reason why it's important, as many of you will know, is that we need to feed a growing world population. Rice, for example, feeds almost a third of the world's population currently, and we need a lot more of it in the next very short period of time. There's about a billion people who are currently malnourished, and so that's not just not getting enough calories, but not getting enough vitamins, micronutrients, and so this is a big challenge for humanity. And the problem is that we don't have enough land to grow the amount of crops that we need unless something else changes. And this slide here just illustrates what's happening in southern Tunisia, where the Sahara Desert is moving further and further north, and therefore reducing the amount of arable land that's available. So you'll all be aware that the population's predicted to continue to grow, and that's not evenly spread across the world. And in fact, the population towards the end of this century is predicted to drop in Asia, at the top here, this yellow graph, and Europe and the Americas. Most of the growth, the major growth, is going to be in Africa. And so this represents a sort of a very important challenge that we have to deal with. So in terms of agriculture, what we can think about is effectively two kinds of strategies for crop improvement, because crop improvement is something that we need to do in terms of yield, in terms of quality, resistance to pests and pathogens, and so on. And the way breeding strategies could be classified, if you like, is into conventional breeding and GM technologies. And what I want to do is sort of compare and contrast these two approaches. If we want to be very targeted in either strategy, we need to understand a lot about the genetic basis of these various traits that I've listed on the left-hand side, and that's a major challenge. Once we know key genes involved in traits, then we can select for them using conventional breeding, molecular assisted breeding, or using a direct introduction of those genes into crops through GM technologies. The way in which conventional breeding has been carried out typically is through combined technologies of hybridization, mutation, selection of phenotype, and by genotype. So with the advent of molecular biology, there's been the introduction of fairly precise molecular techniques to follow genes associated with traits that are important for example, crop yield or resistance to diseases or pests or pathogens. So I want to sort of just say a little bit about the conventional breeding, which has been hugely beneficial, particularly since the Second World War, though it did begin a lot earlier than that, about 10,000 years ago, when Neolithic man stroke woman started to collect seeds, started harvesting wild species for their benefit, and unconsciously selecting those individual plants which had benefits for their purposes, mostly for food. But then there was a kind of a step change in the 1800s through the work of these two guys, which I'm sure everyone's very familiar with. So Charles Darwin, of course, very eloquently explained how species were most likely not just planted on the earth by God, but actually can and were therefore immutable, but actually do change. But of course, he didn't understand the basis of that. And then Gregor Mendel on the right-hand side was also able to quantify the inheritance of traits across generations. Again, he didn't really understand, of course, the molecular basis of that, genetic basis of that, but we have a much clearer idea of both of these things now. And this is one of my favorite sort of agricultural pictures. Does anyone kind of see what's peculiar about this painting? Sorry? It's either they're very tall or these are hobbits. Yeah, exactly right. So ancient wheat varieties were very tall, and they had all sorts of problems in terms of yield because they would put a lot of carbon energy into stem growth, and they would fall over and lose seed in the soil if it was a sort of a windy, wet day. And so a major step forward was the development of so-called semi-dwarf varieties. So this would be an ancient variety here, and this would be a dwarf, and these would be semi-dwarf varieties of wheat in this case. So this is turned out to be a very important trait, which was originally developed through the work of this chap here, Norman Borlaug, who was an American wheat geneticist and breeder, and he was working during the 1940s and 50s doing mutation breeding. And this slide on the left illustrates so he was working in Mexico, and from 1950 up to 2000, you can see the increase in yields in the varieties that he developed in Mexico compared with others used in the Indian subcontinent. And he received the Nobel Peace Prize. There isn't a plant Nobel Prize for that work. But what you can see from the graph on the right-hand side is that yields have plateaued. So those increased yields that are illustrated on those graphs were partly due to genetics, but also due to input of agrochemicals, so pesticides, fertilizers. But we're now getting to the point where we need to do something else. If we're really going to try to address these challenges that I mentioned at the beginning, associated with feeding a growing world population against the background of climate change, which is limiting the availability of arable land. So, so the key challenges then in this sort of so-called molecular agriculture, we could call it, is to raise the yield per acre, because we're not going to have more land as things stand. We need to improve resistance to biotic and abiotic stresses. We need to improve quality. And underpinning this, if we really want a targeted approach to molecular plant breeding, we need to understand the genetic basis of these important traits. So, so let's look at these two approaches and of conventional breeding versus GM. Conventional breeding has been very effective, and I showed that from the graphs from Norman Borlaug's work. We've seen massive increases since the end of the Second World War. But there are problems in taking this further to develop even better varieties. One problem is genetic linkage. So you could have a gene that maybe gives you bigger seeds, but it might be closely linked on a chromosome with one that confers susceptibility to a disease. And because these genes are so closely linked to each other on a chromosome, they're kind of difficult to separate out by conventional plant breeding, because it's trying to shuffle apart, say, 2 cards in a pack that are adjacent to each other within that 52 card pack. Just probabilistically, it's quite difficult. Many important traits are polygenic. So again, the shuffling of genes to get the right combinations together is difficult if there are multiple genes involved in, for example, yield, to use a crude trait term. Therefore, you know, it's difficult to get the breeding working very precisely, and takes a very long time to get the right combinations of genes. There are also problems with the size of the gene pool for many crops. So, for example, at the beginning of the 20th century in India, there was something like more than 100 different varieties of rice that were commonly cultivated. Now the number of varieties is much reduced to less than 5. And that means that you can't readily introduce new germplasm into your elite varieties from existing varieties. And then, of course, we've got problems of timescale. So it takes about 18 years to develop a new wheat variety through conventional breeding. So all of these challenges are quite problematic. And so we need to think about other approaches. And this is why I want to sort of introduce some of the possibilities around a GM technology. So the way this works is that you have to introduce a gene into some plant cells. Typically, this would be from a different species. So it's a very direct way of introducing a gene that you think is really important for yield or pest resistance or fungal resistance or whatever. And you need to introduce that into cells from which you can then use tissue culture to typically anyway, regenerate plants from cultured cells like this stuff up here. And that sounds okay, delivering genes to these cells and regenerating plants from them. But it's not that straightforward, because not all important crops are that easy in practice to transform. So cereals and legumes are notoriously tricky. You can do it, but it's not easy. Not very high frequency is what I mean by not easy. That regeneration process is often genotype dependent. So if you have an elite variety of wheat, for example, you may not be able to transform that particular variety. So it very much depends on the context of the genome of that particular variety. In many ways, it's more art than science. You have to try different protocols in order to get regeneration to occur. Perhaps a more serious problem is one of identifying genes that are actually going to make a difference if you put them into an organism. Will they actually benefit, in agronomic terms, the crop? So there's a lot of science required, as I'm sure many people will be aware, in order to identify genes that are meaningful in terms of crop improvement. A lot of the basic science is carried out on organisms like Arabidopsis. I know some of us here have been working on them for longer than we care to remember. Very powerful organisms to work with, these so-called model organisms, because what that means is that thousands of people around the world are trying to understand the way in which genes work in that particular organism. There's a huge critical mass of effort, lots of tools available, molecular cell biology tools available to help understand mechanisms of development, responses to stresses, and so on. It's much easier to work in a model organism than in a major crop, and that's because crops are tricky because they're often polyploid. So that means you can't find mutants. Mutants are very powerful because if you knock out the function of a gene and you find it doesn't make, it makes a smaller seed, you know that gene is required for the control of seed size. But if you've got a polyploid crop, you have redundant function between genes on various genomes within the plant. And genomes are often very large and complex. So the genetic experiments that you might need to do to understand gene function in a crop can be quite problematic. As I've already mentioned, many traits are polygenic, which makes it quite difficult to identify really important genes that contribute to yield or quality. And because it's quite difficult to genetically transform a number of these crops, then testing function by, for example, overexpressing a gene or expressing it in different cells is quite difficult if the transformation isn't very efficient. So there are problems. Another kind of problem that we had to deal with when I was on ACRE, of course, was the regulatory process and public acceptance of GM technology. Both of those are serious barriers to commercializing a technology that relies on GM technology. Depends on which part of the world you're talking about. So in North America and in Europe, the regulatory system is very different. And this is quite pertinent to sort of common current discussions about regulation of GM. So in Canada, for example, determining whether something is suitable to be released into the environment in farms depends not on how you generated the plant, but looking at the safety, environmental health safety of the final product. So in other words, you look at the product rather than the process. Whereas in Europe, the regulatory system is focused principally on the way in which the product, the plant that you want to release into the field was generated. And if it used recombinant DNA technology, it falls under the EU regulatory umbrella. And that is a major issue for companies that want to release products into the environment. It's a very expensive, slow process to get through that regulatory system. And in fact, you'll probably know that there are very few examples of commercial GM crops grown in Europe currently. And that's because getting through the regulatory system, which is also subject to veto, I mean, a decision is subject to veto by any of the member countries. And Austria always votes against. So that's a bit of an issue. Of course, public acceptance is another consideration. Many people, just for various reasons over the last 20 odd years, have not been happy with the prospect of eating GM foods. We can discuss why that might be. So we have a protracted regulatory process. In the UK, the advice to government comes through this committee called ACA, which I mentioned before, and in Europe there are other committees that then make decisions that go to the Council of Ministers in Brussels to make a final decision, and that's where the vetoes from different countries can come in. Assuming something's approved, it has to go through a kind of quite in-depth post-market monitoring process. So in applications to put things out into the field, you have to have, as a company, a seed company, for example, very clear a description of how the monitoring of the thing in the field, how it performs, how it might potentially cross with other plants. All this has to be discovered and covered and discussed. And then, of course, there's the whole public debate about, I think personally where it went wrong before was that in Europe anyway, people didn't really see the need for GM technology. They could, go and buy their bread and it didn't have to be GM. They just go to Tesco's or Sainsbury's to buy it, and they don't really see that there's a pressing need to do anything else. So the choice is there to have GM or not, and they chose not, by and large. So what might the benefits, potential benefits of GM technology be? Well, on one level, it introduces genetic variation into a breeding population in kind of the same way that conventional breeding does. I mean, the point of all the crosses in conventional breeding is to introduce new genetic variation. The problem with conventional breeding, as I mentioned, is that you may be doing that from a fairly restricted gene pool. But in terms of GM, there's no restriction on where the target gene comes from. And one example of that, I was talking to David briefly about this before, is the golden rice example, where you can introduce genes encoding enzymes for the biosynthesis of carotenoids, vitamin A in particular, from different species. And you end up with rice grains that will produce their own vitamin A, which in some parts of the world is an important vitamin that's lacking in the diet. Because you're introducing genes directly, irrespective of other genes that might be close to it in a genome, you avoid this problem of linkage drag. So potentially that could be much more efficient if you can identify the right kinds of genes. So this potentially can speed up the breeding process, but not necessarily by a huge amount. I mentioned that it takes about 18 years or so to develop a new wheat variety. Depending on how much genetic information is available on a target gene, you might be able to reduce the time to about 15 years, because you still have a protracted process of backcrossing to ensure stability of the traits, and it still has to go in Europe through, well, everywhere it has to go through a regulatory process. The details of the regulation are in different parts of the world. Now, a lot of GM crops have been grown in the world. I mean, the first ones were commercially released in about 1996, I think. And by 2016, 185 million hectares of GM crops of one type or another were grown around the world, as indicated on that graph up there. Mostly in North America, and South America, China, Australia, some in Africa, not very much in Europe there. And what's grown? Well, the most common crops that are grown as GM crops are soybean, maize, cotton, oilseed rape, and some others at much lower levels. And the numbers that have been, the acreage that's been grown has steadily increased. This is from '96 when the whole technology kicks off, just to 2008. But you can see the examples here are herbicide tolerant soybean, cotton, and corn, and BT cotton and corn. And I'm going to explain why these crops were. These were the traits that were in these crops in a moment. They increased, and they're still increasing. So what I want to do next is just talk about some of the examples and why the following examples have been developed as GM crops. There are good reasons why these ones were chosen. The main reason is that genetically, the traits that I'm going to mention now are simple traits. In other words, they can be conferred on a plant by introducing one gene that has a dominant effect. In other words, it doesn't matter. It doesn't have to be homozygous for it to have an effect. It can be heterozygous, but that single copy is enough to confer the trait. And what we're going to talk about is herbicide resistance, or tolerance, really, I should say, rather than resistance, insect resistance or tolerance, and tomato fruit quality. So these are conferred by single genes, although in more recent years, the genes have been stacked. In other words, you could have a crop that has both herbicide tolerance and insect resistance. So the first example I'm going to talk about is from at this point, people put garlic around their neck and form the sign of a cross. So Monsanto were famous for misreading the popular view of GM technology, particularly in Europe. So they kind of bulldozed their way through the regulatory system. But nevertheless, this is one of the sort of classic examples of a GM crop that is out there, and it's out there in large quantities, particularly grown in the Americas, and that's GM soybean, known as 43-2. They're very exciting names they give these things, and it's resistant to a herbicide called glyphosate. So you can buy it called Roundup. Glyphosate is an inhibitor of an enzyme involved in essential amino acid biosynthesis in plants. The enzyme that it inhibits is not present in mammals, and so it's considered relatively safe because it doesn't inhibit any of your own metabolism as a human being. So what it does is this small molecule inhibits this enzyme called EPSPS, which is in the shikimic acid biosynthetic pathway required for the synthesis of these essential aromatic amino acids, phenylalanine, tyrosine, and tryptophan. And unlike some herbicides, it's a broad spectrum herbicide. It will kill broad-leafed crops, not crops, don't want to kill the crops, broad-leafed plants and cereal and grass weeds. The way it works is that the herbicide accumulates in the meristem, and of course, if you're inhibiting amino acid biosynthesis in meristems, in the growing points of plants, you get no protein synthesis or reduced protein synthesis, and so the meristem stops growing and eventually the plant dies. And it's considered to break down quickly in the soil with a quite a short half-life. So it seemed like the ideal kind of herbicide. So this is the enzyme that's targeted. Actually, this reminds me of a joke. a joke, just an aside. So a guy goes into his local garden centre and he said, I want to buy a litre of the inhibitor of enolpyruvol shikimate phosphate synthase. And the chap behind the counter says, what? He says, I want to buy a litre of the inhibitor of 5-enolpyruvol shikimate phosphate synthase. And the chap says, do you mean glyphosate? He said, yes, I could never remember that name. Anyway, this is what it does. This glyphosate binds to that and inhibits it. Now, you might think if you're inhibiting through glyphosate this enzyme, where's the value in making a GM plant that involves this kind of metabolic pathway? And the answer is that there are mutant forms. You can generate mutant forms of that enzyme. And typically these are selected for in bacteria because bacteria, although mammals don't have this pathway, plants and bacteria do. And it's very easy to screen for mutants that don't bind glyphosate. So you can generate a mutant form of this that you then put in your transgenic plant, and therefore, when you apply glyphosate, the herbicide, the plant's got this copy of a resistant form of the enzyme, and so it can still make the product. So it survives, whereas all the weeds around it will die. So there's a bit of kind of molecular biology fiddling around that you need to do. This enzyme is encoded by the nucleus, but actually functions in the chloroplast. So you've got to target it to the chloroplast. And so it's linked to a chloroplast transit peptide, in this case from Petunia, in this Monsanto example from Petunia. And the mutant form of that enzyme is derived from an Agrobacterium strain. So when you spray with glyphosate, the weeds die, but the soybean crop doesn't because it has this resistant form. And the considered advantage of this is that because it's a broad spectrum herbicide, you don't have to spray multiple times. You can just reduce the number of sprayings you do. So that's the argument. So there we have an example of a trait that's conferred by one single dominant gene. The second example is also a single dominant gene, and this is around another Monsanto GM maize, in this case, rather than soybean, trait which confers resistance to insect pests. And in this particular example, a gene is cloned from this bacterium called Bacillus thuringiensis. This is a species of bacterium that produces a range of proteins that are toxic to particular, not to every, but to particular, depending on the protein, Lepidopteran pests. And these generally are known as Bt toxins, after Bt for Bacillus thuringiensis. They're endotoxins and they disrupt the functioning of the gut of the Lepidopteran pest. So in Mon810, this transgenic line, there's the gene, the Cry1AB gene, which is the gene that encodes one of these Bt toxins, is driven by a strong promoter in the transgenic plant. It's also got an intron from maize, so it's a chimeric kind of gene construct that's gone into the plant, and that intron enhances mRNA stability. And the argument here is that you're avoiding spraying fields with pesticides because the plant is producing its own pesticide. One of the kind of ironies of this story is that there's a lot of public unrest about the use of Bt toxins in plants, but in fact it's perfect. You can even buy Bt toxins in your garden centre and use them as a spray. And although that's not regulated, because you just go and buy it over the counter. Actually, the GM crop is regulated, and it's because in Europe it's regulated on the basis that it was generated through recombinant DNA technology. So this is where you start getting kind of you know, strange outcomes from a particular kind of regulatory process. And I'll come back to that point when we talk about gene editing later. And then the last example, some of you in the audience might remember buying GM tomatoes in Sainsbury's. I certainly do. When I first went to Durham, you could go down and buy GM tomatoes. And these were tomatoes that were modified in their ripening characteristics, which I'm just going to explain. So the ripening of tomatoes is a tightly regulated, genetically regulated process, and it's mediated through this hormone, this gaseous hormone called ethylene. And at a certain point in the development of the tomato fruit on the plant, the ethylene promotes ripening by increasing the carotenoid content, that's the red stuff, conversion of starch into sugars, and wall softening. So it's a very tightly regulated process. Now the problem of leaving a tomato on a plant and harvesting it is that it doesn't travel well. It will taste fantastic, but it's not very good for packaging up into cartons and going to supermarkets and then being sold. So this wall softening process causes post-harvest damage. So what supermarkets do is they harvest early. So that's why you get these sort of yellowish things that don't actually taste of tomatoes in your supermarket, unless you go for the expensive kind of Waitrose version. So the idea here was to use technology that was originally developed based on a lot of work carried out at Nottingham University by Don Grierson, where he identified the mechanisms of ripening in tomato fruit. But it was taken to market by Calgene, which is a Californian GM company. And they produced these flavor saver tomatoes. So what they did was it was understood from Don's work that these kind of enzymes, which digest the pectin between the cells in the tomato fruit, are responsible primarily for the softening of the fruit. So the idea was you could inhibit that softening process, but not affect the other sort of beneficial features of ripening, i.e. colour and taste, by suppressing the activity, the expression and activity of these cell wall softening enzymes, and that's what was done. So you could then harvest the fruit late. They would taste good, they would look good, but they wouldn't be all squishy, and so they would survive much better. And this is kind of what they look like compared with a normal tomato at that stage of development. So that's the third example of a relatively simple, from a technological point of view, technology, relatively simple genetically to engineer, because you're talking about single genes here. The problem comes when you want to modify some other traits, such as metabolic traits. So metabolism in plants, animals, is extremely complicated. Many of you will have seen these kind of metabolic charts, and some of us even had to memorize some of them at one time. But to try to engineer something that's so complicated is quite difficult. And I'll try and briefly explain why. So this would be my simplified metabolic pathway, where a metabolite A is converted by an enzyme into B, which is converted into C, which is converted into D, etc. A might be up here. Don't know what letter it would get through down here, but you get the point. It's quite a complicated process. And so when people were back in the 80s trying to figure out how you could use GM technologies to engineer metabolism like this, it was pretty difficult. The reason it was difficult is, okay, so here's the 3 enzymes that would be regulating this pathway. If you increase the transcriptional activity of one of these enzymes, say E2, does that mean you'll get a faster flux through the pathway if that was originally a rate-limiting enzyme? And the problem is, it doesn't, generally speaking, because as soon as you upregulate this and push the activity of this enzyme, this enzyme then becomes limiting. So when you've got a sort of a really complex network like that, you can't readily engineer it one step at a time. And in fact, these metabolic pathways are coordinated, and they're coordinated by regulators of the entire pathway. I mean, I'm slightly generalizing here, but in many examples, they're coordinated by what's known as a transcription factor, which will activate all of those genes in a particular pathway at the same time in a coordinated way. And I'm going to give you an example of that. So this is relating to oil yield. Oil's a valuable crop. It's important for nutritional purposes, calorie intake for many people around the world, also for industrial purposes as well as lubricants and so on, engine oil. So how could you engineer quite a complex pathway like oil biosynthesis and get crops to produce more oil? And the answer is that there is a transcription factor, a single transcription factor that activates the entire oil biosynthetic pathway just by introducing one gene. And that's a gene known as the wrinkled transcription factor. There are now other examples where this is the case, but I just kind of like this example. So what we've got here is the accumulation of triacyl glycerols, which are the seed storage oils that you find in oilseed plants like Arabidopsis and also oilseed rape. And normally in seedlings, so these are seedlings don't accumulate significant levels of these oils. And that's what this bottom line is showing. But this line going up here is what happens if you transgenically overexpress the single gene, the transcription factor known as the wrinkled transcription factor. So here's an example where, again, putting in one gene can have quite a big effect. You don't have to think about all the metabolic steps in oil biosynthesis that you might have to somehow independently regulate. You just put in a master regulator of that pathway and you can produce more oil. And there are other examples of this, and I'm going to sort of finish my examples talking about Kathy Martin's work, because it's quite topical at the moment. So Kathy is at the John Innes Centre. She works on, well, traditionally she's been working on anthocyanin biosynthesis. And the first example I'm going to talk about is engineering anthocyanins in tomatoes. So the red colour, so many of you know that anthocyanins are the kind of purple pigments that you see in many petals. The red colour in tomatoes, as I mentioned before, is not anthocyanins, it's carotenoids. But anthocyanins, such as you can see in her little basket of fruit here, have a number of known health benefits. They have antioxidant properties and potentially could be quite useful in a food product that was widely available. So from her work, her basic science using antirrhinum, she identified transcription factors that were required for the synthesis of anthocyanins in antirrhinum flowers. And what she did was she introduced two of these transcription factors, one called Rosea, one called Delilah, into tomato fruit under the control of a tomato fruit specific gene promoter. So that just activates these two genes in this case, in tomato. And lo and behold, you get these purple tomatoes. So these purple tomatoes have just been approved in the last week by the US Food and Drug Administration for commercial production. Although this paper, as you can see, came out in 2008. So things take quite a long time to get through the system. Why is this potentially important? Well, what she did in her original paper was demonstrate that mice fed on her purple tomatoes lived longer. That's what this purple line is. Than mice fed on ordinary tomatoes. So and that's what these lines are basically here. So and it's believed that this is through the antioxidant properties of those anthocyanins. So these will be on sale probably sometime soon in the US. I don't know whether you'll be able to get them in Sainsbury's here, but we'll see. The other example I'll talk about from Kathy's lab is using a new technology, CRISPR-Cas9, which is again in the news a lot at the moment. And I'm going to say a little bit about that later. But so this is about again, Kathy's interested in engineering improved nutritional quality in a common crop like tomato, so added value to the tomato crop. And in this case, it's about introducing vitamin D into tomatoes. Now vitamin D is it's a steroid that is generated from a precursor. So this would be the precursor, following activation by UV. So in other words, when you go out and sunbathe, you're increasing your vitamin D levels, and you will know that, I'm sure. There was an interesting study in Glasgow, actually. So this was from Sue Newnham in Surrey University. So she's an expert on vitamin D, and she was involved in a survey for the Ministry of Defence on vitamin D levels in submariners that were, spending 6 months under the oceans in nuclear submarines. So she measured from a sample of submariners of their vitamin D level, and as a control, she just took a random selection of people from Glasgow, because these submarines are docked in Glasgow. And yeah, you know what I'm going to say. Who had the lower vitamin D levels? It was the Glasgow population. True. So if you could find a way of easily, without sort of feeding tablets to people, engineer vitamin D in a commonly eaten crop like tomato, then potentially you're onto a winner. So what Kathy, this is kind of again complex, quite a complex. This is a simplified sterile biosynthetic pathway diagram. Anyway, the key point here is that you get this precursor here converted to this molecule here, which is the molecule that normally under UV exposure would be converted into vitamin D. Plants don't normally do that. Actually, they just push the metabolites all the way through to cholesterol and other sterols. Not much cholesterol actually accumulates in plants. Anyway, what Kathy did was create a mutant in this enzyme here, which converts that into that. And when she created that mutant, what she found was there was a shunting of this molecule here into, well, it accumulated so that it could then be converted by UV into vitamin D. And that's what these data here show. So this is the accumulation of DHC, this metabolite here. Normally, there's quite a lot of it in a in a tomato, and under UV. So there's quite a lot. There's not much of it in a tomato because this pathway normally just shoots all the way through to the bottom. But when you mutate this enzymatic step here, what you do is you get an accumulation of this intermediate DHC. When you treat with UV, which is this dark blue band, the level drops down, and the reason it drops down is because it's converted into vitamin D. So you're getting a metabolic shunt across simply by blocking that next step in the pathway that would normally metabolize DHC into sterols. So this is relatively recent work. It was published just this year. So where I'm going to finish off now by sort of thinking about where things are going in the future. One of the techniques that Kathy used in that last example, the vitamin D example, was CRISPR-Cas9, and you'll all have heard about this because it's potentially valuable in agriculture, it's potentially valuable in medicine. Essentially, what it involves is the targeting of a DNA cutting enzyme, a nuclease called Cas9, at specific parts of the genome. So it's a kind of molecular scissors that's guided by an RNA molecule so that it binds to a specific site and cuts at that particular site. And what that means is that you can target mutations. When Norman Borlaug did his mass mutagenesis to find the semi-dwarf varieties of wheat, it was a completely random process. He used a lot of mutagenic chemicals to do that, and or radiation, but you can't control it. It's a completely random kind of process. I say completely random, fairly random. What this technology allows, and it was developed by these two scientists here, who got the Nobel Prize a couple of years ago for this. It allows you to create mutations in genes that you think are important. Think of Kathy's vitamin D example. Or you can cut the DNA so that you can add transgenes at that particular site. Now, what's quite interesting about this technology is you may get round the regulatory process in Europe, and this is under active consideration by the European Commission at the moment. In the UK, because of Brexit, yeah, I hate to say anything positive about Brexit, but one of the things is that we may not have to follow European regulation. Part of the reason why this technology may get round the regulatory process is that if you create a mutant by CRISPR-Cas9 technology, the genome would look exactly the same as a mutant that was caused by natural means. So you can't police. You could say that's good or that's bad, but you certainly can't differentiate because there's no genetic fingerprint other than the mutation that's been created. And the UK government's discussing this, and when I was on ACRE, which was a few years ago now, we released some advice which made the point that if there's no GM footprint in your crop and you cannot distinguish your edited crop from a naturally occurring mutant, should that not be considered differently to one where you're overexpressing a transgene, for example? So there's much more recent advice now come out of ACRE, guidance on using gene editing for making GM plants. So this is an area that's going to become increasingly in the news, I think. It's interesting, the potentials are quite large. You still need to identify important genes. So this crop biotechnology approach, it's never going to replace conventional breeding, but it could augment it. It offers certain advantages. In other words, you have a broader gene pool from which to identify genes that could be useful. But if you really want to engineer your crops, you really need to understand genetic mechanisms that underpin the important traits. And that's where a lot of the difficult work is. And that's, of course, ongoing in multiple labs around the world. So on that, I'll finish, Durham, and I'm happy to take any questions if there are any. So thanks very much.
For years, so that was really nice. So have we got any questions, please? Richard, the first one I see.
How far are we from putting C4 into a C3 plant?
Well, there's quite a lot of interest in trying to do that. I think we're still, I mean, I think it shouldn't be that difficult because it's evolved independently so many times, but no one's been able to do it yet. And I mean, there's a couple of groups in the UK for sure who are working quite actively on this. Jane Langdale and in Oxford, Julian Hibbard in Cambridge. Hasn't happened yet. So I think what my suspicion would be that there are some key transcription factors that regulate the anatomy, that regulate the metabolism, and you need those in combination together to get the whole thing to work. And while there are, you know, there are mutants that affect the anatomy and there are mutants that affect the metabolism, the key regulators have not been identified, and they certainly haven't been brought together in one plant to try to do that. I think it's probably not that far away. There's a lot of knowledge around the whole system now, but we're just not there yet. It turns out to be more complicated than you might at first thought imagine.
I thought it was very, very complicated.
Just for the sake of this recording, Richard asked how soon we'll be able to put C4 into C3 plants. So if you could repeat
sorry, yes, I'll try and do that. Yeah.
Julia?
In your introduction, you said that 100 years ago, India had 200 varieties of rice, and now there are 5 aged ones. So what is the parallel situation, for instance, in the UK with our crops? How narrowed down are we with the varieties that are most planted?
Okay, so the question is, to what extent are UK varieties narrowed down from what was available 100 years ago? I don't know the exact numbers, but certainly the numbers of varieties that are grown are fewer than they were before. So you've got winter wheat and spring wheat, and within that there are a number of different cultivars that are used quite a lot. But I think what some research groups are trying to do is to introduce genetic variation from wild accessions from the Middle East to a large extent in order to try and increase the gene pool size, the availability of resistance genes of one type or another. So I don't know. I mean, we have quite a few wheat varieties in the UK, but I can't tell you exactly how many we've got, but almost certainly much fewer than we used to have. And that's because the ones that we use have been quite well proven to grow well under UK environmental conditions. Now, of course, this summer was not a typical UK summer, and ones over the next few years, decades, are likely to be somewhat different to the conditions over the previous few decades. So the target is moving. We're going to need to get new variation in the breeding population. So a bit of backtracking, I think.
Yes, I was thinking when you were talking about Africa being a target country, you know, it's awfully.
Well, yes, no, yeah.
distance north to south,
Yeah.
massive range of
Yes.
climates in there. Where do we begin?
Well, yes, and I suppose another question is whether wheat would be the appropriate crop for different countries in Africa. There are lots of local crops that could probably be improved that people are happy to grow. There's a thing about growing Western crops in Africa because there's a sort of a status about that for small farmers, but they're not necessarily the best things to be growing. And so I think one area of importance is to try and work with people that are using endemic crops and get them better. Thanks.
To that, when I came to Edinburgh 50 years ago to work with zero crops, a certain group of cultivars, wheat, barley, oats, and I don't think any of those I have them.
Right. Yeah. I'm sure that's the case. Thanks.
It's like widespread that there are herbicides and insect resistant crops and things like that. Would we be breeding into the environment, or like not breeding into the environment, but like evolving other species that like pests for crops, for example, that just become resistant to BT crops, and then that's just kind of like
Yeah.
a
So the question is whether by introducing a GM crop with, for example, insect resistance, would have an impact on weed species, for example. Is that what you're asking?
Yeah.
Yeah. So this is something that ACRE used to discuss quite a lot. Every time there was a new application to release a crop, it was important to think about the wider economic and ecological consequences. One of the things you have to remember is that wheat, for example, doesn't cross with anything that you would find growing in the countryside. So you've got species barriers there in place. A lot of crops don't overwinter well, so they wouldn't necessarily survive and therefore spread. Wheat is not very good at overwintering, some wheat. So I think there are barriers, but I think this is where the post-market monitoring plan that has to be developed as part of your application to release a new GM crop comes in. So you have to explain how you would deal with any unforeseen issues like that. So it's certainly something that's part of the regulatory process.
So yes.
They do, but they evolve. Yeah, I mean against pesticides you spray on. So this is not specifically a GM kind of issue. It's a battle between insects and whatever resistance mechanism there happens to be. It's not specific to GM.
You think that bettering crops through GM carry information? Is that something that eventually is it going to happen anyway?
So the question is crop improvement going to help the human race, and is it worth doing?
In terms of the population?
In terms of population number. Yeah, it's a good question philosophically, I suppose. I would say there are other things that could be part of the equation. So reducing waste, for example, reducing food waste is one thing. I think, I mean, if you're asking, is the population going to keep increasing anyway? Possibly. And I mean, in Asia, for example, and Europe, it's not going to keep increasing. So the issue really in the next 100 years is really around Africa. Now the question is, do you think it's ethical to not try to provide the kind of food supplies that people might need there? Or do you think, would you say, hands up, it's going to increase anyway, so what, how? You know, I think that's a policy decision really for governments about how they want to deal with their increasing population. But I mean, it's an interesting thought, but I think a lot of scientists would probably want to try and at least provide the options there, and then, you know, governments will decide whether they want to take it up or not. I mean, golden rice, for example, has not been taken up by a lot of governments, even though it could have an advantage for problems associated with vitamin A deficiency, blindness. And that's because they didn't like the idea of GM crops. So yes, it's not a straightforward situation, but I think what as scientists would probably want to do is to provide opportunity, and then policymakers decide what they want to do. So the question is, are there any GM crops currently cultivated in the UK? Not for commercial purposes. There are experimental crops. So, for example, there's new fungus resistance genes in potato that are being cultivated for experimental purposes. And at Rothamsted, there are some oil crops being cultivated for experimental purposes, but not currently for commercial growth.
Very interesting talk. There's been some significant changes of heart amongst some prominent anti-GM activists. I'm thinking of Martin Linus, who 10 years ago now probably, said in the New York Times he made a mistake and wrote a book about this. Is it your perception that opinion has changed to see the advantages of this more?
Yeah. So the question is whether opinion has changed amongst groups that were quite anti-GM maybe 10 years ago. And my perception is yes, actually. So Greenpeace, for example, have stepped back from a very strong anti-GM stance. My feeling is that it's for probably two reasons. One is that there's a need for more food and better quality food. That's one thing. The other is that I think there are some environmental benefits to the use of these products. So, for example, I mean, I think a good example is the Bt resistance. This is a way of avoiding spraying stuff and, fuel associated with planes flying over fields. That doesn't have a great environmental benefit, net benefit. And so if the plants can produce these things themselves, I think it's probably a good thing. So yeah, I think there's a subtle change. I mean, there are still people that are quite vocal against genome editing, but I've always found it interesting that when it comes to medicines, people are much happier to, like insulin is a recombinant protein, right? People are quite happy to inject that into their bloodstream, but not eat a GM tomato. So, but I think things are changing. I think what's important is the debate, the public debate, rather than the mistake that companies like Monsanto made, where they just assumed everyone would think they were great and would welcome their technology with open arms without really discussing the implications of it. So I think it's really important to do that, to have those discussions. And I think what we see now is, yeah, a softening of that anti-GM stance, because there are potentially benefits and need. It's all about need. If you don't need it, if you don't need a firmer tomato, for your breakfast, why would you bother? But I think if there's a need, then I think that changes the game, and that's why medicines, are more readily accepted, because you you need it.
Just to pick up on that point about need, don't you think we need GM crops in Europe more than ever now, with the farmers need to cope with high prices of fertilizer, increasing restrictions on the use of pesticides and herbicides, and having to adapt to climate change and testing?
I mean, I do. I think yes, because spraying, especially with energy costs now, I mean, the act of going around with tractors or planes spraying stuff is going to become, farmers don't have that huge profit margins as it is. And of course, the energy required for producing these things. So that combined with climate change, as you suggest, I think is a good reason why this is quite important. And I think will happen in due course, so long as, I mean, of course, when the technology was new back in the 80s, people were quite uncertain about what the implications of it might be. And in Europe was established this precautionary principle. We don't want to take undue risks. But I think these things have been grown now for nearly 30 years, more than 30 years. And I think they're fairly well understood in terms of the general technology. I think you always look at a given crop on a case-by-case basis. But I think what we should be doing, where I would hope there might be a change in the regulatory process, is to look at the product rather than the means by which you produced it. Because we understand so much about the means by which you've produced it. There's really no risk with that. You just have to look at whether the product itself is safe or not, which is what happens in North America. And I think that's a much more sensible, balanced approach.
Well, I think one should have a point, and unfortunately, we now have an anti-GM King Charles III on the throne. We also have an anti-GM Scottish government.
Yeah, so the point being made was we have anti-GM royalty and Scottish government. Yeah, yeah. I think I don't know the extent to which King Charles will influence things. I mean, it's quite difficult when you get to that age to change the way you work. But he did make some comment that, when I'm in this role, I'm going to have to behave differently. I think what I would hope is that that the scientific advice would be taken and the government would make a decision on that basis rather than listening to him. In Scotland, yeah, I mean, yeah, it's going to be a local decision, isn't it?
Yeah. And thank you again to Professor Lindsey for a fascinating talk and answering all of those questions. Wonderful. And just before we close, I'd like to invite people who aren't members to please join. You need to go via the website. There are some leaflets here. You have options. It's only £5 if you're a student. You have options to get the journal and the Climate Society newsletter. So if you aren't a member, please do join, and thank you again for being such an attentive audience. See you again in the future.